Hello and welcome back to my channel, Explaining Biology Clearly. Today we're going to talk about how the function of different organelles in a cell are interrelated with one another, so the relationships that they have together. We're going to look at how a protein is made and how it's eventually secreted or released from the cell, and how the organelles involved in that process work together, if you like, to do that. So on the left hand side of the page down here I have a picture of the nucleus. So the nucleus is here, and around the outside of the nucleus is the nuclear membrane, sometimes known as a nuclear envelope. Along the nuclear membrane you'll notice these gaps, so these are the nuclear pores. They allow substances to move in and out of the nucleus. Inside the nucleus is another organelle called the nucleolus. So there's two organelles that we're going to have a look at. These are membrane-bound organelles, so we're talking about a eukaryotic cell. Do see my video on eukaryotic cells to learn more about their structure. So we'll also talk about the rough endoplasmic reticulum and ribosomes and the Golgi body and the cell membrane, and I'll come to those in a moment. So first of all, we'll have a think about the nucleolus and its function. So the function of the nucleolus is to make a molecule which is called ribosomal RNA, also known as rRNA. So it makes this substance, and this moves out of the nucleus into the cytoplasm, the jelly-like substance surrounding the nucleus and filling the cell. It moves out of the nucleus into the cytoplasm, and in the cytoplasm it attaches to proteins and forms an organelle called a ribosome. This is not a membrane-bound organelle. It doesn't have a membrane round it. It's just an organelle. So ribosomes can be found free in the cytoplasm, and they can also, in eukaryotic cells, be attached onto a membrane-bound organelle called the endoplasmic reticulum. So the endoplasmic reticulum is a series of tubes, if you like. They're all interconnected to one another, that is originally attached onto the nuclear membrane but spans and spreads out into the cytoplasm. So the ribosomes can be free in the cytoplasm, or they can be attached onto the surface of this endoplasmic reticulum. If they're attached onto the surface, then this is actually the rough endoplasmic reticulum, as opposed to the smooth endoplasmic reticulum that doesn't have any ribosomes on its surface. So nucleolus makes ribosomal RNA that moves out into the cytoplasm, where it forms the ribosomes, either free in the cytoplasm or attached onto the rough endoplasmic reticulum. The function of the ribosomes is to produce proteins. So let's start with that. So the ribosomes produce proteins. They're the site of protein synthesis. Let's just add up on there then the nucleolus. This produces ribosomal RNA. So the ribosomes are made of ribosomal RNA. Okay, now let's go back to the nucleus. So what do we find inside the nucleus other than the nucleolus? So we find DNA. And DNA in the eukaryotic cell are linear strands. Can be tangled up inside the nucleus, forming a mass called chromatin. So I've drawn a little bit of chromatin there, made up of these linear strands of DNA. Now, what is DNA? DNA is a molecule, it's a type of nucleic acid, and it contains the code, the instructions for proteins. One of these instructions, one of these sets of instructions is called a gene. So a section of DNA that codes for one protein is called a gene. And that gene is carrying the instructions for one protein. That gene is copied or transcribed, as the proper word is, into a piece of messenger RNA. The DNA, you'll notice, is very large. It can't leave the nucleus, so that instruction needs to leave the nucleus in order for the ribosomes to make the protein. And the only way that instruction can leave the nucleus is if it's made into a smaller molecule of messenger RNA, and that still contains the code for the protein. So messenger RNA then leaves the nucleus and enters the cytoplasm, and it attaches on to a ribosome. So hopefully you can start to see the way that these are linking together. So let's just add that to our description over here. So messenger RNA has the code for, let's put a protein, and it attaches to a ribosome. The nucleus contains DNA, and genes are copied into messenger RNA. Okie dokie. So once that messenger RNA is attached onto the ribosome, in this case on the rough endoplasmic reticulum, but it could be one of these ribosomes that are free in the cytoplasm, the instructions on that messenger RNA are used to make a polypeptide. This is the initial form of the protein. It's not yet ready to carry out its function. It's not been folded up yet. It's not yet ready. In fact, I've drawn it folded up, haven't I? Let's not draw it. Can I change it? I don't think I can. So it's not yet folded into a protein. So it's called a polypeptide. Now that polypeptide there gets internalized into the rough endoplasmic reticulum, and it then gets packaged. It gets packaged into a vesicle. So we draw a vesicle. A vesicle is a piece of the membrane surrounding that polypeptide. It's like a membrane sac containing the polypeptide. So it's just a little section of the rough ER that has been cut off, and inside it is that polypeptide that's just been made. So one of the things that the rough ER does is it packages polypeptides. Packages them. It then transports the polypeptides. So that vesicle made of the rough endoplasmic reticulum membrane will then move to the next organelle we're going to talk about, which is the Golgi body. So here is the Golgi body. It's a series of membrane discs arranged like a stack of pancakes, and that vesicle from the rough ER attaches on to the Golgi body, and so therefore the protein, the polypeptide, becomes internalized in the Golgi body. It's inside the Golgi body. And so what the Golgi body that does then is it modifies the polypeptide. It modifies it, and what that means is it folds it. It might fold it into a secondary, tertiary, quaternary structure. It might add prosthetic groups like iron in the case of hemoglobin. So it modifies the polypeptide. So it's producing the protein, the final product that can carry out a particular function. Once that Golgi body has done that, it's modified the protein, the protein then gets packaged again into some Golgi body membrane. So a bit of the membrane pops off containing the protein. So there's a vesicle that's being produced once again. So let's just add what the vesicle is. So a vesicle is a membrane sac, and it contains a protein produced by either the roughy R or the Golgi body. If that protein is an enzyme, then it contains an enzyme. The protein is an enzyme. So it's called a lysosome only if that protein is an enzyme. That vesicle then moves to the cell membrane. So here's the cell membrane here. I've done a gap deliberately, I'll show you in a minute. So the vesicle moves towards the cell membrane, and when it reaches the cell membrane, it fuses with it. The vesicle's made of membrane, the cell membrane's made of membrane, so they can fuse together, and then that protein is then released, it's secreted. And that process of fusing and secreting is known as exocytosis. Exocytosis. So the vesicle fuses and secretes, or fuses with the cell membrane and secretes the contents. Fuses with the cell membrane and secretes its contents to the outside, to the tissue fluid around. Right, I'm going to stop there. I hope that helps you to understand how to explain the answer to a question on how function of organelles is interrelated. Just go through the function of each and it will fit together. Please like and subscribe to my channel and check back for more videos soon. Thanks for listening. Bye.